And now let's move to Marty Yang, that is our second speaker today. And Marty is a PhD student in Michael Greenberg's lab at Harvard Medical School. And there he studies genetic variation to understand how transcription factors control cis regulatory function in the context of native chromatin. So Marty, when you are ready, you can go ahead.
Hopefully you can see my screen now.
Yes,
Great.
it's perfect.
Great, thank you for the very kind introduction and for the opportunity to speak today here. I'm happy to share research that I conducted as a graduate student in Mike Greenberg's lab, where we took a genetic approach to try and dissect the function of enhancers across the genome and the function of a class of transcription factors, in particular, the AP1 set of factors in detail. So just to set a little bit of context, as many of you are well aware, the mammalian genome comprises millions of cis-regulatory elements that control gene transcription. But the focus of my talk today will be these gene distal enhancer elements because they're highly important, we think, for regulating cell type specific transcription. So just for some context, enhancers are relatively short stretches of DNA sequence, and they bind combinations of typically 4 or 5 transcription factors via these short DNA binding motifs that I've depicted here. And they function to potentiate transcription at target promoters. And they do this by recruiting the pre-initiation complex and transcriptional coactivators via these long-range groups to target and activate gene transcription. Now, while promoters are proximal to transcription start sites and often ubiquitously accessible across many cell types, enhancers, on the other hand, prior to the binding of transcription factors, are actually occluded by nucleosomes or histone optimers in most cellular contexts. And in fact, they become functional only upon the receipt of specific extracellular stimuli, which can induce the expression of, say, stimulus-responsive transcription factors, or in other cases when cell type specific transcription factors are expressed in the right combination and at the right levels. So a relatively common observation that I think has been emerging for a number of fine mapping studies in recent years is the fact that many putative causal SNPs that they're finding often occur outside of any known transcription factor binding motifs. And if we think that the binding of transcription factors are really these critical switches for enhancer function, one might naively assume that this result is a little bit surprising. What I'll argue today is that this suggests that features outside of core transcription factor binding sites contribute significantly to the binding and function of transcription factors. So if we take a look at this green transcription factor depicted here, which doesn't contain a mutation within its core binding site, what might some of these other features, sequence features, be that dictate a loss in binding for a factor of interest? So you can imagine a number of possibilities, and I'll just mention a few of them here. For instance, you could imagine that disruption of motifs for putative collaborating factors, if the collaboration is required for the binding of the factor of interest, this could be in some cases sufficient to block binding for a given transcription factor without a mutation within the core binding site itself. Secondly, we have an increasing appreciation that the sequence context in which the motif itself is found can often influence binding affinity, and in particular, sequences adjacent to core motifs can modulate features such as DNA shape within the core binding site itself. And then lastly, as I mentioned at the start, these transcription factors need to bind their enhancers within the nucleosomal context. So you can imagine that simply altering nucleosome positioning could alter the affinity for a transcription factor when it needs to access the histone optimer. So there are, of course, a number of approaches by which one might be able to study the determinants of enhancer function. And in particular, the one that we sought to utilize was to leverage naturally occurring variation that exists between inbred mouse strains. And our reasoning for this was that this could effectively function as a large-scale mutagenesis screen for enhancers and also for transcription factor bound regions in the genomic context. And this has several advantages, notably the first one, which I've depicted here, is the fact that from a single genetic cross between these mice, we can survey about a tenfold greater degree of genetic divergence than would be possible by comparing maternal and paternal alleles in a typical human sample. And secondly, because these mouse strains are relatively high quality genome assemblies, and we've had them for several years now, this really enables what I'll argue is a highly quantitative comparison of, say, enhancer function across alleles. So with this general framework, one additional question we wanted to ask is, since enhancers across different cell types often exhibit very distinct patterns of enriched motifs, can we find a class of transcription factors that might control enhancer function in a cell type invariant manner, or in other words, across a large number of cellular contexts? And for this, we were inspired by work actually from Matt Morano from a number of years ago, in which he and others had performed measurements of chromatin accessibility across a number of human tissues and cell types. And the main finding I want to point out here is the fact that when they looked at the total numbers of SNPs, the numbers of SNPs that were imbalanced, or in other words, caused a skew in chromatin accessibility, and the proportion of these two, and they did this across a number of annotated transcription factor families, what they found was that the AP1 motif, when it was mutated, this was the class of transcription factors that was most likely to result in an allele-specific change in DNA-seq signal. And so this suggested to us, and this is one of several pieces of evidence, that we think that there might be a relatively pervasive role for AP1 in regulating enhancer function across the genome, irrespective of the cellular context. So something that's, I think, particularly striking about AP1, and it's a set of factors that Mike's lab has studied now for several decades, is the fact that the expression of the AP1 TS at both the mRNA and the protein level is elicited by RAS-MAP kinase signaling. But importantly, this can occur upon the receipt of actually distinct stimuli across different cell types. So much of our lab is interested in understanding how neuronal activity is converted through the action of AP1 TS to go on to affect synaptic plasticity. What I'll go on to talk to you about today is actually how a distinct set of stimuli, in this case, the addition of growth factors in fibroblasts, can elicit a highly distinct cellular adaptation, principally wound repair. And importantly, a central question we want to address as a lab is how these ubiquitously expressed transcription factors can go on to control cell type specific genes that have distinct functions within these different contexts. So as I mentioned, there are a number of systems in which we can study the function of AP1, but in particular, we chose to work with a relatively reductionist system, which are primary mouse embryonic fibroblasts, or MEFs. For one reason, they're highly homogeneous, at least relative to the types of studies that our lab typically does in the mouse brain. And secondly, by growing the cells in low serum for extended periods of time, we're able to synchronize the cells or arrest them in G0, the cell cycle, which can allow us to control for cell cycle dependent effects on, say, enhancer function. And then lastly, by serum reintroduction at high levels, we can trigger the activation of the AP1 set of factors and study their function when they're maximally expressed. But also by virtue of cells re-entering S phase, we can really also study the function and state of enhancers across two different stages of the cell cycle as well. So as you can imagine, there are a number of ways to go about this problem. And the approach we took to really dive deeply into sequence determinants of enhancer function was to simply survey a very, very large number of alleles for each regulatory element of interest. And to do this, what we did is we crossed C57 black 6 female mice to a diverse panel of inbred male mice. And some of these included a number of male mice from wild-derived lines. So these are going to be highly genetically divergent from black 6. And so we generated F1 hybrid fibroblasts from all these crosses and others that I have not shown here. And from these, we performed a number of allele-specific measurements that we thought were representative of cis regulatory function. So we assayed for chromatin accessibility to define putative cis regulatory elements across the genome. We also quantified in allele-specific fashion a number of histone post-translational modifications that were representative of both enhancer and promoter function. Thirdly, we performed allele-specific measurements of binding for AP1, for TEAD, which we think is a set of factors also that are stimulus responsive and that we think function as a putative collaborating factor for AP1 in fibroblasts. And then lastly, we also mapped the binding of CTCF, which contrary to these other transcription factors actually principally binds at insulators across the genome. And then lastly, we also performed measurements of gene transcription to assess whether changes in enhancer function ultimately go on to regulate differences in gene expression. And then we used high chip as a method to link enhancers to other active enhancers or promoters, because often these elements act at great distances from their targets. So I thought one way to be able to summarize the nature of our data set would just be to show what our data looks like in aggregate. So what you can hopefully appreciate here is that we're plotting here H3K27 acetyl levels. So this is generally known as a proxy for active cis regulatory function. And we're doing this in this plot at all proximal elements. So these are going to be principally gene promoters. And each individual point here represents a separate promoter. And we're plotting the signal from in each case, from the black 6 allele and the corresponding paternal allele for each F1 hybrid. And what you can hopefully appreciate is the fact that the vast majority of points fall along the diagonal here. So this suggests that for most enhancers in our data, or for most promoters in our data set, they contain SNPs that ultimately don't result in a functional change in histone acetylation. And hopefully what you can also appreciate, and we quantify this here on the bottom, is that a number of points end up showing either a black 6 or a paternal specific skew in their signal. And these are sites we're considering allele specific. So if we do this across a number of histone modifications of interest, as well as for CTCF binding levels, we can start to get a sense of how these distinct features of chromatin might differ from one another across the genome. And for instance, we can start to ask questions such as whether a promoter and enhancer associated modifications, whether they differ in their patterns of allele specificity. So in this case, we did find that promoter associated histone acetylation, as well as this H3K4 trimethylation mark, are less likely to show an allele specific skew in signal than enhancer associated modifications, such as gene distal K27 acetyl or the mono or dimethylation of H3K4. And this is, of course, consistent with a number of prior studies that have suggested that enhancers tend to functionally turn over at a greater rate than promoters. But most of those studies have typically been done looking at more divergent sets of mammals. So with this sort of large scale resource, one can imagine asking a number of questions, but just for the sake of time today, I'm going to try to highlight a couple of vignettes that really leverage the allele specific nature of our data set, which I think is a distinguishing feature. So one question we want to ask is, what are the types of variants, or what are properties of variants that are most likely to predict a change in enhancer function? And then secondly, we want to take a deeper dive into the AP1 set of factors because we think they're critical for enhancer function. And we want to assess how they might alter enhancer chromatin state. And also, we want to better be able to define how AP1 binds specific enhancers in distinct cell types. So to address this first question about the nature of variants that are associated with changes in enhancer function, we started simply by assessing the number of SNPs and indels present at enhancers and promoters across all our F1 hybrid lines. And we've classified these individual enhancers and promoters based on whether they show or do not show an allele specific skew in histone acetylation. And what you can hopefully appreciate from this plot is that well, a number of features, but the one in particular I want to highlight is the fact that allele specific sites for both enhancers and promoters exhibit on average a greater number of SNPs and indels than sites that don't show a functional change in histone acetylation. And I think one thing that's particularly interesting from this plot is the fact that this change does not appear to be a global shift. So in other words, while this difference is significant, what we're observing here is that the number of genetic variants alone is not a sufficient predictor to be able to reliably classify whether a given enhancer or promoter is likely to exhibit allele specific activity. So secondly, what we wanted to do was to examine the relationship between the total numbers of SNPs and indels and the fold change in histone acetylation levels between the active and inactive alleles. And we did this for both allele specific enhancers and promoters across the genome. And what we observed from this particular analysis was that we could observe only at enhancers and not as clearly at promoters, a scaling in which greater levels of histone acetylation changes between alleles can be observed as you consider enhancers that have increasing number of genetic variants. So this suggested to us really two things. The first of which is that with the way we're performing the allele specific mapping, we are indeed able to observe quantitative differences in histone acetylation, especially at enhancer elements. And secondly, to us, this also is consistent with the model in which, at least for enhancers, that SNPs within the data set appear to each additively contribute to changes in enhancer activity. So given these two pieces of data, one hypothesis we wanted to assess is the extent to which the location of a SNP or indel might be a better predictor of whether it's likely to affect function than merely the number of SNPs overall. And so this is what this analysis is attempting to get at. So we've plotted here the frequency of SNPs and indels across all enhancers in our data set. We've done this by centering all these plots on the summit of chromatin accessibility. And shown here are the running averages for the frequency of variance between sites that show an allele-specific skewing signal in black and those that show similar levels of histone acetylation between both alleles. And what we observed here is relatively central enrichment within about the core 100 base pairs or so of enhancer regions for disruptive SNPs and indels. And I'll just remind you that a typical enhancer comprises an accessible chromatin window of about 150 to 200 base pairs. So it's really this core 100 base pairs where we observe this particular enrichment. And then secondly, if we ask, if we want to assess specifically the types of variants that are sufficient to disrupt the enhancer function, and in this case activity, we were able to repeat the same analysis, but we looked specifically at sites that contain a single SNP or indel. And these are likely to be SNPs or indels that are causal in their ability to block enhancer activity. And we note a similar length scale of enrichment of SNPs and indels in this case. So with this sort of general dive into how variants might disrupt one particular feature of enhancer function, which is this histone acetylation mark we were interested in, we also wanted to take a deeper dive into the function of AP1 using this F1 hybrid approach. So one particular aspect of AP1 that I hadn't mentioned at the start of this talk that I wanted to highlight is the fact that our lab and others over the last several years have been interested in what the function of this class of transcription factors might be. And we got some clues into this from a co-immunoprecipitation experiment that we performed a number of years ago in a prior study using FOS EGFP expressing MEFs. And when we performed this co-IP, what we observed was actually an enrichment of a number of peptides corresponding to subunits, core subunits for the canonical switch SNF or BAF complex. So this suggests to us perhaps that a principal function of the AP1 TFs is to recruit switch SNF or BAF to enhancers across the genome. However, we still lacked an ability to directly test the requirement for AP1 and perhaps this interaction with BAF for the function of enhancers in the genomic context. So to address whether this interaction and also other features of AP1 might be required for enhancer function, we of course reason that one logical approach would be to do this with gene knockouts. And so I'll just remind you that the AP1 transcription factors comprise both gen homodimers and also phosgen heterodimers. And they bind this palindromic AP1 or TRE motif that was largely identified from in vitro binding assays. And since then, we've gained an appreciation that these motifs are actually highly enriched in enhancers. And really, to make a long story short, we and others had knocked out several of these AP1 transcription factors and really had noticed, we were a little bit surprised by this, relatively minimal effects on the function of most enhancers across the genome. So this suggested that we really needed a distinct approach to be able to define, for instance, their sequence binding preferences in chromatin. And given the fact that we had this F1 hybrid data, we thought to take a relatively targeted approach to be able to address this question. So we reasoned that instead of perturbing these transcription factors in trans, we could identify a subset of loci across the genome that are bound by AP1 and that contain a single AP1 site within the presumably enhancer region in which we observe a mutation or a variant that disrupts that is predicted to disrupt binding of AP1 TFs at this locus. So these are AP1 bound allele pairs in which it contains a single AP1 site that's disrupted by a predicted mutation. And we were able to identify about 1,000 or so of these, about 1,300 or so of these allele pairs. And we first simply asked at these active FOS-bound enhancers, what the signal for FOS binding, which is an AP1 family member, looks like between the alleles in which the motif is intact versus lost. And what you can hopefully appreciate is the fact that mutation of a single AP1 site results in at least an aggregate of quantitative loss in AP1 binding, but itself is not sufficient to predict the loss in AP1 binding. And I'll discuss this in greater detail in some subsequent slides. So with that, we can start to ask what also occurs at these corresponding alleles to different features of chromatin state. So even with a relatively modest aggregate loss of AP1 occupancy, we noticed a relatively substantial and complete block in chromatin accessibility, but also a loss in histone modifications associated with enhancer priming here, and also enhancer activation. So this suggested to us that loss of AP1 binding that's driven by cis-actin mutations, at least for us, this picture suggested that it results actually in a block in enhancer selection, meaning that we observe both the loss in this priming associated modification, as well as this modification associated with active regulatory elements. So from here, we wanted to ask simply what other sequence features might better predict the loss in AP1 signal than core motif mutations alone. So in our initial search for AP1 binding sites, and this was done in black 6 fibroblasts, one feature we found was that nucleotides beyond the core motif itself, which I've underlined here, were often highly stereotyped in our motif searches. So it suggested to us that in the context of chromatin, that these flanking nucleotides might actually contribute to binding specificity. But how can we assess this using our allele-specific data to really test whether these additional nucleotides are required? So we reasoned that if we looked at active FOS-bound enhancers, and we subsetted to look at the set of sites that exhibit an allele-specific skew in FOS binding, we would expect to find an enrichment of SNPs, specifically at nucleotide positions that are required for DNA binding affinity, but not at nucleotides adjacent to that. And that's precisely what we see here. So we observe a relatively strong signature of SNPs within the core binding site for AP1. And we noted as well a relatively modest enrichment of SNPs within these flanking base pairs. So it really suggests to us that these flanking nucleotides can actually play a role in contributing the binding affinity for AP1 within the chromatin context. And then lastly, I just wanted to walk you through one final analysis we've done with this sort of more refined binding site for AP1. We sought to utilize our variant data to understand how AP1 might collaborate with other signal-dependent factors to bind, and whether this might explain some of its cell type specificity. So just to remind you, enhancers are typically occluded by histone octamers prior to transcription factor binding, and typically the binding energy from any given transcription factor is thought to be insufficient to overcome this nucleosome barrier to access underlying DNA. And one emergent model that's been proposed over the last several decades is a collaborative competition model in which posits that the coincident binding of several transcription factors is required to elicit either nucleosome eviction or remodeling. And with this particular model, this coincident binding can only occur when the motifs are positioned on the same side of the nucleosomal dyad or on the same face of the nucleosome. So the dyad itself is the point of symmetry within the nucleosome. And within this model, it's thought that the motifs must occur within 75 base pairs of each other in order for this collaborative competition to be able to take place. So can we specifically look at AP1 in our data set and assess the extent to which AP1 might utilize this mode of collaborative competition to access its binding sites in the genome? So what we did was we examined a set of FOS peaks across the genome. And importantly, this set of FOS peaks had an intact motif for AP1 on both alleles. And in this case, we've centered our plots on this shared binding site that's intact on both alleles. And we simply asked at this point how SNPs and indels are distributed at allele-specific versus shared sites relative to this intact motif. And what we observed actually was an enrichment of SNPs within about 50 base pairs or so of this AP1 site that's shared. So this suggested to us, or at least is consistent with the collaborative competition model, because the motifs, the mutations themselves are enriched within 75 base pairs of this intact site. And importantly as well, this distribution is non-uniform. So in other words, the enrichment of SNPs does not occur at a highly stereotyped position, which would be what you might expect to see if AP1 was largely engaging in direct cooperativity rather than this indirect collaborative competition model. And I'll just note quickly that we observe a similar pattern of SNPs when we perform the same analysis looking at TEAD binding sites. So TEAD we think is a collaborative factor for AP1 and fibroblasts, and we observe a similar pattern suggesting also that TEAD is largely functioning through collaborative competition. When we consider CTCF on the far right, we observe a somewhat distinct pattern, and in particular, we notice a much more restricted length scale for disruptive SNPs. And this is consistent with prior studies that have suggested that CTCF, at least in the context of insulators, can bind relatively independent of collaborative binding interactions. So just to summarize what I've shown you today, we performed extensive allele-specific mapping using F1 hybrid fibroblasts in mice to identify thousands, in this case, of enhancers that exhibit either strain-specific activity or strain-specific patterns of transcription factor binding. And we think this could be a valuable resource for the genetics community to be able to perform downstream analyses as well, such as deep learning type analyses. We also found that SNPs within the core 100 base pairs or so are the ones that are most likely to disrupt at least enhancer activity, and we've observed this across thousands of loci within our study. We've also observed through cis-acting mutations that the first time that we think AP1, that a loss of AP1 binding really results in a block in the selection of enhancers across the genome. And then lastly here, we defined a number of what we're calling non-canonical sequence features. Really, there's a sequence features beyond core AP1 sites that we think contribute to binding affinity. And this includes nucleosomes flanking core AP1 sites, as well as something I haven't mentioned today, but we also think that partial motifs also, partial AP1 motifs can also contribute to binding affinity in chromatin. And so with that, I just want to thank the folks who've contributed to the study, namely my advisor, Mike Greenberg, as well as Tommy Verbuchen, who was a former postdoc in the lab, is now running his own group at Sloan Kettering. And then secondly, I wanted to mention that this entire work was a collaborative effort between myself and a former student in Mike's lab, Emmy Ling, who's now in the genetics department here. And also we received help from Chris Calley, a former technician in the lab. And then I've just included my contact info here in case anybody would like to reach out to discuss further. As well, I should mention that our work has been pre-printed on bioRxiv, so it should be, additional details should be available there as well. And of course, thank you to everybody here for your attention.
So thank you so much, Marty, for this very interesting talk. Remember that you can type your questions in the Q&A box. And maybe in the meantime, I can just start with one question.
Sure.
So at the beginning, when you were showing correlations between paternal and maternal different expression, like, can some of these features be explained to some extent by the maternal or paternal strains? Or like, do you see differences between the different strains?
Right. So
especially like comparing paternal strains specifically.
Right. So if I'm understanding the question you're asking is, do we see strong evidence that, say, the nuclear environment or transacting effects might contribute to differences in any of the chromatin features that we'd assayed? Right. Yeah. So one particular feature that we looked at is actually histone acetylation signal on the X chromosome, because I neglected to mention that we performed these studies only in male F1 hybrid fibroblasts. So they'll and because of the way we set the breeding, having a black 6 female in all cases means that these F1 fibroblasts will have a single X chromosome that's from the black 6. And so from that, we can attempt to quantify histone acetylation levels without the need for allele-specific mapping, which of course, as many of you are aware of, has a number of potential biases. And I haven't shown the data here, but we observed essentially very few instances of enhancers on that X chromosome that show a significant skew in histone acetylations, depending on the particular F1 hybrid context in which we're assaying them. So it really suggests to us, at least as far as we can measure, that transacting effects were having relatively minimal effects on histone acetylation. And then conversely, I think that implies that we can more directly study the function of cis-actin mutants, which are principally the ones in this case that we're interested in assaying.
Mm-hmm. Great, thank you. Let's see. Okay, in the meantime, I'll just go with another question. So at the beginning, you also said that you had two different conditions in your experiment design.
Right.
Could you explore some specific effects between the two conditions?
Right. So just to remind everybody, we serum starve all our fibroblasts prior to performing experiments, and then for half of them, for half the conditions, we re-stimulated with high levels of serum for 90 minutes. And what we observed in terms of at least for the enhancer landscape across the genome, is that the vast majority of enhancers don't exhibit at least a qualitative change in their histone acetylation patterns or their chromatin accessibility patterns, but a small subset of them, about 10% of enhancers across the genome, show an acute, and we think it's stimulus dependent, increase in chromatin accessibility and also histone acetylation. So it suggests that these enhancers are becoming dynamically active only upon the receipt of, in this case, an extrinsic stimulus. And then I think one particularly interesting feature of this set of enhancers that we've started to dive a little bit deeper into detail is the fact that these enhancers are typically occluded by nucleosomes, actually, so we can detect relatively high levels of H3 signal at these sites prior to serum stimulation. So we think that this small subset of enhancers across the genome might allow us to, in particular, examine how AP1 TFs, because their expression pattern also is induced by the stimulation, how they might be able to interact with the nucleosome and give us a better sense of how some of these pioneering events that AP1 might contribute to in a broader set of enhancers across the genome. We think that this specific set of enhancers might lend us some insight on how that interaction might be occurring.
Okay, thank you so much, Marty. So if there are no more questions, maybe it's time to close. So thanks everybody again for attending the Variant Effects seminar series today. And just make sure to be back for next month's seminar, which will be on the 3rd of May, if I'm not wrong. And we will have Deborah Sobreira and Antonio Molica. Thank you all, and thank you, Sean and Marty again.
Thank you.